Okay, this is Unit 7. We're going to be looking at Mendelian genetics and incomplete dominance, all right? And so this will get you into the first section of your note set, and in 7.1, we're looking at the history of genetics, a short amount of history, and focusing on the work of Gregor Mendel. All right, so let's consider a lottery scenario, right? So say I'm going to roll a dice, 6 dice, and I'm going to roll them one at a time. And what I'd like you to do is write down in order the numbers you think that I will roll from the first dice through the 6th dice. So take a moment, write those down. Okay, so let's say that these are the numbers that you have selected, all right? And let's look at the odds of you actually matching the numbers in the actual order that I roll them, right? So the odds for the first number being a 5 would simply be 1 out of 6 because there are 6 sides to a dice, and so the chance of you rolling, of me rolling a 5 is 1 out of 6. And so if we consider what is the odds of you selecting the second number being number 2 from that second dice being rolled, it's still also 1 out of 6. And what we'll find is that each number individually that we have selected, there is a 1 in 6 chance that we have actually picked the correct number. So the big question then is, what is the overall odds that you would actually have the correct series of numbers in the correct order, right? And we simply multiply these factors, the chance, 1 out of 6, together, and it comes out to be 1 in 46,656 times. And so this would be a 0.0002% chance of you actually selecting these numbers, right? So the idea is that in genetics, we're going to look at probability. The probability is that the traits or the phenotypes that we see are a result of the genotypes being passed on, and so what are the odds of obtaining those genotypes? All right, so generally genetics is looking at a variety of things that are associated with heredity and also the variation of those organisms. And we know that that information, the genetic information, is carried on chromosomes, right? And these chromosomes, like this chromosome here, right, carries the information in genes. So each little band or each little location on this chromosome could be a code for a particular protein. So a sequence of DNA can be transcribed into a sequence of RNA, which can then be translated into a protein. And this is the central dogma of biology. And so we'll get into this central dogma idea in the next unit of material. Chromosomes occur in pairs, and the genes that they contain are also kind of paired up in homologous chromosomes, right? One from mom and one from dad. So these new combinations of genes will occur because of the sexual reproduction process. The formation of the gametes kind of recombines the DNA, and then the selective fertilization or union of the sperm and the egg, the haploid cells to form the diploid organism, is what really drives this new unique combination of information from 2 parents. What we need to also keep in mind is that these homologous chromosomes are going to have a code for a specific protein, and this one will also have a code for that same type of protein. What might be different is what is ultimately expressed, like the flavor of that gene might be slightly different from one parent to the next. But we'll look into more of that idea of genes and alleles in just a little bit. Heredity is the process of passing that on. And Gregor Mendel, who's an Austrian monk, right? And so he, from his work, uncovered a pattern of how traits can be passed from generation to generation. That it's not just random accident that these things happen, that there's actually an underlying process, right? And he did that by studying the pea plant. A little background behind Mr. Mendel. He was born in 1822. He was the son of a farmer, and he was actually an ordained priest. And so he studied botany and learned how to use and apply the scientific method. And so he decided to look into or investigate this passage of traits from one generation to the next through a pure line of pea plants. And so before his time, people were actually thinking that inheritance of traits from your parents was simply a blending of their traits. That like whatever made up your mother and what made up your father is actually just kind of distilled out and then dissolved and blended into what makes you up. Much in the same way that if we take an apple and a banana and put them into a blender, we get an apple-banana smoothie. You have the flavor of the apple and the banana kind of mixed together. But that's not what Mendel saw. What Mendel saw in his plants was that some plants don't necessarily show a blending of traits. And so there must be something else going on as far as how traits are moved from parent to offspring. So he studies these 7 traits of the pea plants. Most people are familiar with the flower color and seed texture and seed color. I think most people have done Punnett square work with that before. But he also did work with the pod shape, the flower position, the pod color itself, as well as the overall height of the pea plant. Right, and so it's these 7 traits that he studies in this lineage of pea plants that helps him determine the patterns of inheritance. So he publishes his results in which he establishes 3 major principles of inheritance, and we'll mention those as we move through material in this unit. But the thing of it is, he couldn't repeat this work because on accident he kind of had selected a plant that reproduces asexually, and so you do not get that recombination of the genetics from one generation to the next, and so he could not confirm his results in the subsequent plant that he tested. If he had only selected a sexually reproducing organism, then we might be a little bit further ahead in our understanding of genetics. But fortunately, in the early 1900s, some researchers uncovered his work and applied his concepts to other sexually reproducing plants and confirmed his findings. All right, so he was actually the first biologist to actually use mathematics to help explain his results in a quantitative way. So from this, he could predict the concept of genes, that genes occur in pairs, and that one gene of each pair is present in the gamete cells from either parent. All right, there's some general terms you need to know as we move through things, and we'll mention them again as we cover them. But the idea of gene and genome, you should have a good understanding of that. Alleles and locus. So alleles are these 2 genes that will then occupy the same position on homologous chromosomes. So maybe it's like the allele that codes for hair color. That would be found on a similar position, a locus, on one chromosome from a parent, as it would be found in the same or similar location on the homologous partner on the other parent's chromosome. All right, we should also know the idea of homozygous and heterozygous. Homozygous meaning that it's of the same, and heterozygous meaning that we have 2 different alleles or genes present. And then dominant and recessive genes. Dominant alleles or genes are going to be those that kind of overshadow or seem to express over the top of a recessive one, and a recessive one is one that's again being masked by the dominant. So the only way you would see a recessive allele being expressed in the phenotype or the appearance of that organism would be if you had a homozygous recessive set of genes. If both genes from either parent were both recessive, then you would see that recessive trait. All right, so also genotypes and phenotypes, I've mentioned that kind of a little bit here. The genotype is really the genetic makeup, and the phenotype is the actual physical or manifestation of those genes. So it can include both what we get from the genes as well as the environment's effect on the development of the organism. And then when we get into Punnett squares, we'll talk about monohybrid crosses, where we look at just one trait and cross the alleles from either parent. And then the generations that we look at, the parent generation would be the first cross that's done. The offspring from that would be your F1 or first filial generation, and then the second filial generation would be the cross of F1. And then as far as basic flower anatomy, you really need to understand where Mendel was pulling the pollen and depositing it from. So he would choose like one flower of a particular like color or tallness or arrangement, and he would grab the pollen from the anther here, right? So the stamen at the top, the anther is what contains the pollen, and he'd take like a little paintbrush, and he would brush the pollen off onto the paintbrush here, and he would then deposit it onto the stigma at the top of the pistil of another flower. Right? He wouldn't self-pollinate a flower because then he wouldn't. He was trying to see like how traits were being passed on between two different parents to see what how the traits would play out. If we'd simply self-pollinate, then he's taking the same recombinant code from the same source, and so he would take the pollen from one flower and deposit it onto the stigma of another flower. Okay, so let's look at Mendel's experiment. All right, so he took male purple flowers, the pollen from purple flowers, and deposited it onto the stigma of female of another purple flower. So we acquire some of the pollen, we deposit it onto the female, and we then generate this F1 generation, right? And so he had two purple flowers, and these were true bred, meaning that he had made sure that each generation that he was going to pull from had resulted in purple flowers. And so in this crossing, he found that the F1 generation resulted in all 100% purple flowers. And so the thought was that these traits are inherited from your parents. All right, pretty simple idea. All right, so what if we did this with white flowers? All right, so the same process is set up. We take a true breeding white flower, and then we're going to take the pollen from one, and we're going to deposit it into the female of the other. And the result of our F1 generation here is all white flowers. Right? So this kind of followed his understanding that traits are passed on from parent to offspring generations. So what if we were to actually cross the purple flower with the white flower? What would be the result? All right, so that would be his next round of testing. So we take the pollen from purple flowers, and we deposit them onto the white flowers to pollinate them. And the F1 generation results in, well, surprisingly, 100% purple flowers. And so Mendel had to think about this for a little bit, and he came to the conclusion that whatever the trait is, whatever the gene or packet of information that codes for purple must be dominant over whatever codes for white. That if I crossed two individuals that have like two different traits, like this, and as far as flower color, that the purple flower color is a dominant trait over the recessive white trait. All right, so the follow-up question then for him was, what do I do? What happens if I were to cross the F1 generation with each other? What might be the resulting offspring there? All right, so we're going to take a couple flowers from the F1 generation. We're going to run them through the same experiment. So we're going to acquire some pollen from one flower and pollinate another. And remember, these had a white parent, right? So if we then run through to the F2 generation, what we find is that 75% are expressing the dominant trait purple and 25% white. So what do we learn from this? He learned that the offspring will not always resemble their parents, and that these traits can actually skip generation. All right, so what's interesting about this is that Mendel then studied these 7 traits and recorded those as well. And he then documented the ratio of how these things played out. It comes out that they're all nearly 3 to 1. Right? And so that means that this is not an accident. This is not a coincidence, that there is a pattern, there is a method through which these genes, these alleles, are passed from parent to offspring. We've found that genes are like sections of DNA that create proteins. Right? They're located on the chromosomes, and they're inherited from their parents, right? And so they come in dominant and recessive forms known as alleles. And by definition, an allele is kind of like an alternative form of a gene. So on the image here, we have a chromosome from dad and a chromosome from mom, and say that would be the locus of the flower color gene or allele. And we can see that dad's providing a purple allele, the dominant form of it, and the mother is providing the recessive in this case for this individual. And so we use letters then to abbreviate the alleles. In this case, we use the capital letter P for the dominant and lowercase letter p for the recessive. genotype is this genetic makeup of an organism, so we're looking at what two alleles it might have. The gene for purple flowers, the dominant gene for purple flowers, is represented by a capital P, right? And that would also be the same if it's provided from the other parent. And so this one would be known as being homozygous dominant, where we have a combination of two dominant alleles. Right? If we have the gene for recessive white flowers on each parent, and those both get passed on to the offspring, then the offspring would then be carrying a homozygous recessive set of these alleles, of these genes. All right, and then a heterozygous setup would be where we have one dominant allele with one recessive allele. And this is what we would term being heterozygous, a combination of one dominant and one recessive. All right, so it's very easy for us then to look at how the genotype plays out to the phenotype. Right, the phenotype is a physical expression of that gene. Sometimes it's not as obvious as like the color of the flower. It could be other things that are built internally to that organism and how it's expressed and used in their body. Right, but the phenotype then in this case is purple and versus white flowers when you have this combination of either a dominant allele present for the purple color or the recessive allele present for the white color. Mr. Punnett looked at probabilities and the resulting offspring, and that's what so it's a genetic tool that we use to predict the offspring probability, and they're very easy to work with. There's many different methods as far as their application to determine probability. There's some shortcuts out there that they can learn about. I try to avoid going over the shortcuts at this point because sometimes people try to apply the shortcut when it doesn't really truly apply to the individuals that you're using. So I usually just have people walk through the typical setup and run of a Punnett square. We're working with monohybrid crosses, meaning that we're going to only investigate one trait when we cross a couple individuals. All right, so the first thing we want to do is first consider the genotypes of the parent generation or the individuals that we're going to cross together. All right, so we're going to be crossing a homozygous dominant with a homozygous recessive pea plant. So we would want to consider the genotypes of each. So big P for the purple homozygous dominant flowers, and little p for our homozygous recessive. All right, so we're going to take those genotypes of the parents, and we know that these pairs, these allele pairs, have to be broken and will be carried into the gametes that they produce. Right, so the only possibilities really are for the gametes to get one of either one of those alleles from the parent that produces the gamete cell. So we'll go ahead and run these into our Punnett square. And because we're just doing a monohybrid cross, we only have to consider really 4 boxes in our Punnett square here. All right, so we're simply going to carry then or fill across those that we have in the rows. So our dominant, our big P's will be carried across the row that they're found in, and then we'll also then drop the lowercase or the recessive alleles from the other individual down the column, and the resulting combinations in the boxes represent the possibilities or probabilities of the offspring from those individuals. All right, so in this case, what is the probability of having purple flowered offspring? Okay, go ahead. 100%. What would be the probability of if they have one offspring that has purple flowers, of them having a second offspring with purple flowers? 100. It would be 100%, right? Because no matter how we cross these guys, remember every single possibility is 100% purple as far as the phenotype that will be expressed, right? 100% of their offspring will be heterozygous. So just like with a dice roll, remember when we roll the dice, to pick the number correctly from the first roll, it's 1 out of 6 chances. But to actually get correct 2 numbers in succession, we need to multiply the probabilities, right? So it'd be 1 out of 36 to get 2 numbers in sequence correct. So same thing can be applied here if we look at successive generations or successive offspring. when we find the first offspring will come out purple, but we multiply it in the second offspring, 100% times 100% is still 100% chance. It doesn't diminish over time. Also, be careful to, we'll discuss this as we move forward with probabilities, but there's a difference between events that are mutually exclusive and those that are not. So we'll talk about that as we move forward, though. All right, good. So take a minute and run this Punnett square, and if you could, respond by listing the genotypes, the numbers and types of genotypes we see, the phenotypes and their number, and also what's the probability of us from this heterozygous cross with a heterozygous plant of getting a white flowering pea plant. Okay, so we have the genotypes figured out and the numbers of them. What about the phenotypes? Three will be purple and one will be white. Okay, and then what's the probability of having a white plant?
25%.
It is 25%. So, question, we're going to go another one.
Oh, yeah.
Wait. So we have our first offspring, and it ends up being a white flowering plant.
Yes.
Right? What is the probability of us having a second white plant?
100%. Wait, with that one?
Well, from the same parents.
Oh.
So we had one offspring that is a white flowering pea plant. What is the probability of us having a second that is also a white pea plant? Yeah. You had 1 in 4 chance of having a white flowering offspring, right? That's been done, but to have 2 in succession, you need to multiply the possibilities. Yeah, so it'd be 1/16 or 0.065, but that'd be decimal form, 6.25%. All right, so there are 2 major principles that we need to consider with Mendel at this point. One, the principle of dominance, that one allele will mask another, and when it does, that means that must be a dominant allele in play, right? And we'll see that show up when we do the parent generation, or P generation, is the first crossing of individuals, right? The resulting offspring from that cross would be the F1 generation, and if you want to take those offspring and cross them with each other, then that would be the F2 generation, right? The principle of segregation, it also is that when gametes are formed, those alleles, those hereditary factors, the pair of alleles that your father has, or the pair of alleles that your mother has, those pairings will be split into separate gametes to create you, or the offspring. So each sex cell receives only one kind of gene. They don't get both alleles in the same gamete, right? Either they get the purple allele or the recessive allele if we have a heterozygous individual. Right, so these are the 3 major principles. The third one will show up a little bit later on. Okay, so we're going to close today looking at autosomal recessive inheritance. Autosomal recessive inheritance. Right, so this falls in line much like what we see happen with the pea plant coloring of the flower. Right, that in order to have white pea plants, right, you have to have both recessive alleles in order for that to be the case. The same thing applies when we look at autosomal recessive inheritance. Right, it's where that we're looking at the autosomes, so we're looking at not the sex chromosomes, we're looking at the first 22 homologous pairs. Right. And so this is going to be caused by alleles that are found on those non-sex chromosomes. In order to be afflicted or have the disorder or have the disease or have the trait, you have to have both chromosomes that make up your genome will have to have the recessive allele. Right? So just like to have a white flowering plant, you had to have little p. Same thing here. A couple examples. Sickle cell anemia. That's a disorder found on chromosome 11. And the alleles or genes that code for cystic fibrosis are found on chromosome 7. So these are 2 examples of autosomal recessive inheritance. So the sufferers of these autosomal recessive have to get a recessive allele from each parent, right? They have to get 2 recessive alleles in order to be afflicted. So if we take the Turners, right, who are heterozygous, right, and we go ahead and we consider the possibilities, right, offspring 1, if it gets both dominant alleles, 1 from each parent, it's just going to be a healthy individual, right? It'd be homozygous dominant. If we consider the second option where we get 1 of the dominant alleles and 1 of the recessive alleles, then that would be a carrier. They will not have the symptoms, but they have the possibility of passing on the recessive allele to the next generation of offspring. And then the third option, obviously, is if they pass both of their recessive alleles to their offspring, then that person would have the affliction. That individual would have the disease. Okay, I guess I should have put the crying one over here, but I don't know. It just 3 clip art on Google, right? All right, so a human case of this would be with cystic fibrosis. And we understand that Mendel's idea of probability and inheritance works for the passing on of this possible affliction through the autosomal chromosomes. All right, so it's a lethal genetic disease. It afflicts mainly Caucasians, and it's caused by a mutant gene carried by 1 in 20 people of European descent, which runs you to about 12 million people or so are carriers, or have the recessive gene. Right? 1 in 400 Caucasian couples will both be carriers of cystic fibrosis, and 1 in 4 children, that's the probability, will have it. Right now, you may know individuals that suffer from this. I had a pretty close friend that passed away about 10 to 12 years ago from cystic fibrosis. She was like 32 years old, and it's all about affecting transport in tissues, primarily like trying to the cilia that help move the mucus out from your lungs and up your trachea and out, so you don't like drown in your own mucus. That's really what kind of happens. They just can't get the mucus out, and so usually some therapies include like percussion on the rib cage or on the lungs and stuff for them. They'll do that. Seriously, they'll do percussion usually one or two times a day to help break up and loosen the mucus so they can cough it out. But as the disease progresses, it becomes worse and worse, and they get infections, usually pneumonia, and that's usually what ultimately ends their existence. All right, so the inheritance pattern then, if we consider the fact that 2 parents could be carrying the recessive gene, we can do big C, little c for heterozygous, and if we cross those heterozygous carriers, the chances of them having a child with cystic fibrosis is 1 in 4, or 25%. All right, so our Punnett square lines up with what we kind of see statistically occur with cystic fibrosis. Now, remember that these are just probabilities. So consider the fact that there's no guarantee. This is simply an expectation, and it's really the highest probability or highest case or frequency of it to occur, at least for this autosomal recessive disease or disorder. So the reason that we might want to consider these probabilities is that if you're thinking about maybe starting a family, it might be nice to know the family lineage of different disorders. Right? Because we know that there is a very strong genetic link between an individual and their overall health. So it's kind of an important thing to know so you can prepare for what is a possibility. All right. Currently, the most recent statistic is that 1 in 29 Americans is a symptomless carrier of cystic fibrosis. All right. So take a minute, and we have Maria, who is heterozygous and healthy, so big H, little h, and Jeff, her husband or whatever you want to consider him to be, suffers from recessive cystic fibrosis, so little h, little h. Run the Punnett square on them. And they want to know what's the probability of them having a healthy child, and then what is the probability of them having a homozygous dominant child. Okay, this should not take us very long to develop, so we'll go ahead and get this thing sketched in. All right, so we do our cross between our two parents, and this is our resulting Punnett square. So what's the probability of them having a healthy child? Miley? 50%. Yeah, 50%. Remember, just because you're a carrier, it does not mean that you have the affliction, right? It just means you're carrying one of the alleles, one of the recessive alleles. Right? It does not mean that you suffer from the symptoms. Right? And what's their probability then of having a homozygous dominant child? Go ahead, Case. 0%. Yeah, there's no chance for them to have a homozygous dominant child. Right, so the best scenario is that they're going to procreate and have some carriers. All right. So another thing that you might come across when you're having to do Punnett square work is a pedigree. All right, and pedigrees are pretty simple to construct and pretty simple to understand if you know some simple ideas about them. All right, so we're going to do a simple pedigree between Maria and Jeff. All right, and so usually the female is going to be represented with a nice round circle because circles are cool. I have no idea why, just usually circles. Right, so this will be Maria. Right, and this square can represent Hef.
We're Spanish now.
All right, you can put the names in the boxes or outside the box. It doesn't really matter. All right, so if they decide to procreate, all right, we would connect them with a line, and we would continue their lineage to this pedigree. But we also want to include the genotype of Maria, right? So she's a carrier, big H, little h, and Jeff is afflicted, little h, little h. Right, let's say then they decide to have a child. Right, so we're going to show that in this pedigree that they have a child. So the link between Maria and Jeff is now going to be extended into the creation of this offspring. And let's just say that they have, I don't know, let's say they have a daughter. What are we going to name this daughter?
Hannah.
Paul.
Oh.
We'll call him Panna. Combination between Paul and Hannah. How about that? Panna. All right. Is Panna afflicted or not?
Yes.
Yeah? Okay. So she got the recessive allele from mom, the recessive allele from dad, and she's afflicted. All right. Now, what we also should consider is we usually will also shade in those that are afflicted in our pedigree. Right? So Jeff will be shaded in, and Panna will be shaded in our pedigree. And this is really a very simple, like scaled down pedigree between two individuals, but it's something that you may come across, and also something else that you might see in a pedigree is that those that are carriers would be half shaded in. Like you don't necessarily have to see that, but it usually helps you kind of track what type of inheritance disorder you might be looking at, right? So this one, quite obviously, is a autosomal recessive inherited disorder because everyone that has the genotype of little h is afflicted. Yeah. Also, if we would look at the probabilities or percentages of offspring, we would find that in most cases, with enough people present in the pedigree, that we would line up with if these were the originating couple in the pedigree, that we would see 50-50 play out for their subsequent offspring, right? And then we have to run crosses for the next generation in the pedigree to determine what's going to happen there. Okay. What if we didn't know? What if we didn't know the results from Panna? Right? What if we didn't know that? What would we know at least for sure?
Lowercase h?
Yeah, it will. It'll have at least one recessive allele because it has to, from the father, it has to get either little h or little h. That's an absolute certainty. Yeah. So we wouldn't know. I mean, we could end our pedigree by, question mark little h. We don't know if she's a carrier or if she's afflicted. Yeah, that's a possibility too. If they're asking you to consider all options with the pedigree of between these two individuals. Yep. Okay, or between three if we include Hannah.